Hey people, it's NIyas713. This is from the Daily Mail. Chinese scientists develop gene therapy that delays aging in mice and extends their lifespans by up to a quarter, claiming it could one day be used in humans. A new gene therapy has been shown to reverse some of the effects of aging in mice and extend their lifespans by up to a quarter. Chinese researchers use CRISPR-Cas9, a powerful gene editing tool, to inactivate a gene called KAT7, which is a key contributor to cellular aging. An injection of a viral vector encoded for KAT7 reduced what is now known as cellular senescence, the end of cell division which organisms need to grow and replenish tissue. Although experiments were conducted with mice in the lab, the experts think their findings might one day contribute to similar treatments for humans. They believe that there is a lack of investigation on the intervention of genes to treat aging and age-related diseases. The specific therapy they used and results were world first, and co-supervisor of the project a specialist in aging and regenerative medicine from the Institute of Zoology at the Chinese Academy of Sciences. In the lab experiments, all mice lives were extended by 25% on average. Now, for people who understand on a human basis, so imagine the average age is, say, 80. That means that the average human lifespan goes up to 100. These mice show after 6 to 8 months overall improved appearances and grip strength, and most importantly, they have extended lifespans for about 25%. Say study author Professor Ku Jing from the Institute of Zoology at the Chinese Academy of Sciences told Reuters. The team of biologists used the renowned genome editing tool called CRISPR-Cas9, which is used for making precise edits in DNA. CRISPR has been likened to a pair of genetic scissors, allowing for tiny snippets of DNA to be removed and replaced. For this study, the method was used to screen thousands of genes for those with particularly strong drivers of cellular senescence. Cellular senescence is thought to impair tissue function and predispose tissue to disease development. Despite a few previously reported aging-associated genes, the identity and roles of additional genes involved in the regulation of human cellular aging are yet to be fully explained. Researchers identified 100 senescence-promoting genes out of around 10,000, and Cat7 was the most efficient at contributing to senescence in cells. Cat7 is one of tens of thousands of genes found in the cells of mammals. Researchers inactivated Cat7 in the livers of the mice using intravenous injections of a lentiviral vector. Lentiviral vectors have been described as vehicles for gene therapy in mammalian cells. Lentivirus is simply a type of virus. Intravenous injection of a lentiviral vector encoding Cat7 reduced the proportions of senescent cells and pro-inflammatory cells in the liver, and extended the health span and lifespan of aged mice. The results suggest that gene therapy based on inactivation of a single gene may be sufficient to extend mouse lifespan. Cat7 depletion reduced cellular senescence, whereas Cat7 overexpression accelerated cellular senescence. We just tested the function of the gene in different kinds of cell types in the human stem cell, the mesenchymal progenitor cells in the human liver cell, and the mouse liver cell, and for all of these cells we didn't see any detectable cellular toxicity, Ku said, and for the mice we also didn't see any side effect yet. Despite this, the method is a long way from being ready for human trials, according to Professor Ku. It's still definitely necessary to test the function of Cat7 and other cell types of humans, and other organs of mice and other preclinical animals before we use the strategy for human aging or for health conditions, she said. She hopes to be able to test the method on primates next, but it will require a lot of funding and much more research first. In the end, we hope that we can find a way to delay aging even by a minor percentage in the future. Okay, I'm reading this diagram. So what is CRISPR and Cas9? CRISPR is a tool for making precise edits in DNA discovered in bacteria. The acronym stands for Clustered Regularly Interspaced Palindromic Repeats. The technique involves a DNA cutting enzyme and a small tag which tells the enzymes where to cut. Okay, by editing this tag, scientists are able to target the enzyme to specific regions of DNA and make precise cuts wherever they like. It has been used to silence genes, effectively switching them off. When cellular machinery repairs a DNA break, it removes a small snip of DNA. In this way, researchers can precisely turn off specific genes in the genome. The approach has been used previously to edit the HBB gene responsible for a condition called I don't know what that letter is, thalassemia. Anyway, I hope you've enjoyed this. Please comment, rate, share, and subscribe. Peace. If you'd like to support this channel, leave my GoFundMe in the description. If you'd like to follow me on Instagram, I'll leave that there as well. If you'd like to buy one of my t-shirts, I'll leave that there as well. Peace.